All right, so today we are going to talk about non-Mendelian inheritance patterns. If we're going to talk about non-Mendelian inheritance patterns, we should probably start with reviewing what Mendelian inheritance is. You're all experts in that now. Then we will discuss some non-Mendelian patterns, including maternal effects, epigenetics, and cytoplasmic inheritance. I think this is going to be a fun one today. This is one of my favorites. The learning objectives are then to be able to recognize patterns as non-Mendelian, to distinguish between maternal effect and cytoplasmic inheritance. Those are often confused with one another, so it'd be important to be able to distinguish between those two, to explain how X inactivation leads to mosaic phenotypic outcomes, and to predict inheritance patterns for non-Mendelian traits, similar to what we've normally been doing with our Punnett squares, but with a little extra pizzazz thrown in from the non-Mendelian angle. So let's start by reviewing what is Mendelian inheritance. Typically, when we think about Mendelian inheritance, we're thinking about the action of genes directly within the organism that has those genes, right? The expression of genes in the offspring is directly influencing their trait, right? If they have an allele, they have a phenotype associated with that allele. Although there's one instance where that breaks down that we'll talk about today, and that is the maternal effect, where your phenotype does not depend on your own genes, but rather the genes of your mother. There's another sort of classic Mendelian concept, which is that genes are passed unaltered generation to generation, right? Your mom and your dad have sets of alleles, you get those alleles, you pass some of those alleles on to your children, and that the mutations contained within those alleles will affect the activity of that gene directly. But there are cases where you can have transient chemical modifications to the DNA called epigenetic inheritance. And this can transiently affect the activity of those genes regardless of whether or not there are mutations in those genes. Another thing that we think about when we think about Mendelian inheritance is we think about genes obeying law of segregation, right? That you only inherit one of two possible alleles from a given chromosome. There are cases where you don't have chromosomes lining up neatly on the metaphase plate, and that's with cytoplasmic inheritance. That's going to be all of the DNA found in your mitochondria. And then finally, when crosses involve more than one gene, we assume that they obey law of independent assortment, right? That you have alleles from two different genes, you have an equal probability of inheriting this dominant or recessive of this allele or the dominant recessive of this allele. And the violation of that we've already talked about, which is linkage. We're not going to go into that again today, but the basic idea, these are all non-Mendelian, the basic idea is that when you have linkage, right, you have these biased inheritance patterns. You don't necessarily have equal probability of inheriting two different alleles of two different genes. If they're next to one another, they often travel together, as we've already discussed. So we'll go through each one of these other than linkage, and I think we should just get right into it with maternal effect. All right, so this is where the genotype of the mother and the genotype of the mother alone determines a phenotype of offspring. So this will be a rare instance where the offspring has an allele, and it does not matter what the offspring has. It only matters what their mother had. And the reason is that this is caused by an accumulation of gene products in the early embryo. Okay, so what does that mean? So one thing to keep in mind is that the early embryo isn't always making its own genes. As an embryo first forms, early stages of cell division, it's actually not activating its own genome yet. It is instead relying entirely on stuff provided from the mother. So embryos, or the embryo, I guess, itself, let's go embryo, I like that better. The embryo doesn't make its own genes for a long time. Well, a time at least. Instead, it relies on mom's genes. Okay? So what does that look like? When you have an egg that's forming, this will be an egg. That egg is surrounded throughout development by a series of cells. These we'll call nurse cells. And these nurse cells are making a bunch of stuff, and they're pumping it into the egg. Okay? So these nurse cells are providing protein and nutrients and mRNA to the egg, which means that variants in the mother's genome can cause a phenotype in the egg. Okay, so we have a situation where if you think about who's contributing what, we've got a large egg full of nutrients, lipids, proteins, messenger RNAs, and you have a tiny little sperm, right? The sperm's going to come and deliver genetic material, pretty much nothing else. The rest of it is all in the egg. Now, it takes several cell divisions for the fertilized egg and zygote to actually make its own genes. So for a long time, it is doing all of its divisions and its growth using the stuff that's already present in the egg, right? So there's a window here where if you have an mRNA or a protein from mom that has a variant in it, that variant can cause a phenotype in the early embryo. And it will not matter what the embryo's genome has because they're not making anything from that genome, right? So the embryo gets off to a sluggish start. It's not making anything. All of the mRNA and protein for the early cell division is coming from whatever was deposited by the mom into the egg. So what that could look like is if we have an egg here, and we have a nurse cell. This will be a nurse cell. This will be an egg. Okay, and let's say we have a genotype of 2 dominant D alleles. The nurse cell is going to put those gene products that have dominant D alleles into the egg. Right, so the mom is DD. The egg is going to be the D phenotype. Because no matter what the egg has as its genome, it's going to have the dominant D allele from the mom in the egg. Okay, you can also have a situation where the nurse cell could be heterozygous. Well, now the egg is going to be getting both the dominant D allele and the recessive D allele. So mom is heterozygous. The egg is going to then be also the D phenotype, the dominant D allele. But if the mom is homozygous recessive, the egg can only get the recessive D alleles. So if mom is DD, the egg is going to be recessive D phenotype. So it could be that the egg over here has in its own genome the dominant D allele, but it's not going to make it for a long time. It instead only has the protein products and mRNA from the mother that are the recessive D allele. So just to reiterate, it doesn't matter what genotype the egg is only the mother's genotype. Does that make some sense? Right? The egg has its own genome. It's not making anything from it. So you're kind of stuck with whatever mom was able to contribute. That's the essence of maternal effect, right? Not a lot of things are maternal effect, but there are cases where this happens. And I'll give you a classic example. It's the one used in the textbook, which is snail shell coiling. We'll come back to that in a second. So example, classic example, snail shell coiling. All right, so the shells of snails coil in one of two directions. They can be dextral, which is basically a right-handed coil, and they can be sinestral, which is a left-handed coil. Think about kind of chirality of molecules and that kind of thing. It can twist this way or it can twist that way, but they are not the same. And this coiling direction is determined 100% by the genotype of the mother. All right, so the parental genotype or the offspring's own genotype these do not matter for snail shell coiling. It only depends on the genotype of the mother. Let's go through an example. We can have a dextral female here with two dominant D alleles. Dextral is dominant. Or we can have a sinestral male. And when they come together, we have all dextral offspring. And they're all dextral because the mom has dominant D alleles. Right? This actually is just straight Mendelian genetics. On the other side, though, we have a mom that's homozygous recessive and a dad that's homozygous dominant. And here, they're all sinestral, right? The offspring have the exact same genotype, heterozygous, right? The difference is this one, the dextral, had a mom with the dominant D allele. And this one, the sinestral, had a mom with the recessive D allele. Does that make sense? So the nurse cells, in this case, are able to put that dominant D allele into the egg. That leads to this type of snail shell coiling. Whereas this mom had the recessive allele only. Therefore, during early cell divisions, they ended up having a sinestral snail shell coiling instead. Okay. We can take that a step further. Let's take these F1 generations, right? Remember, these are the same genotype, but different phenotypes. Ooh. Okay, right? heterozygous, heterozygous, but this one's all dextral. This one's all sinestral, okay? Now, if you have an individual that's heterozygous, has a dominant D allele, but they're sinestral, right, this would indicate that mother was DD. They can cross to one another and produce an F2 generation. You get, in this case, a 1 to 2 to 1 ratio of genotypes, just like you'd expect, but every single one is dextral. Because every mom in this cross is going to have a dominant D allele, right? So you have this weird sort of dissociation between genotype and phenotype where your brain really wants it to be classical Mendelian genetics, but the phenotype is only depending ultimately on the mother's genotype, right? So you have sinestral moms, you have dextral moms, they all have the same genotype, and then they all lead to dextral offspring. Does that make sense, or is that confusing? A little confusing? Okay. The thing to keep in mind then, the genotype is irrelevant. Right, all that matters is that their mom, their mother, was heterozygous. Right, they are going to be, you can think about it, their phenotype is going to be their genotype of their mother. That's another way to think about it. Now, you're probably wondering how in the world would an allele deposited in a gene by the mother affect snail shell coiling? And the answer is detailed a little bit better in the textbook. But ultimately, these D alleles control the orientation of the mitotic spindle. And during early phases of cleavage in the embryo, you have 2 cells, right? When they become 4 cells, there's different places where those 4 cells could be, right? You could have the 4 cells pop out over here, like that, right, with next cell division. Or, like that, let's say. Or you could have them pop out over here during that cell division, and so on and so forth, right? That individual cell division and orientation of those planes propagates all the way through to the ultimate coiling of the snail shell during development. So that early cleavage plane difference can affect this phenotype much later on in the snail. The textbook gives a better, more eloquent description of that, and you don't need to know the details of it, but those early cell divisions can have a lasting influence, especially in invertebrates. All right, so with that, we can go to a point solutions question. This one for extra credit. This will be session ID 333442. 333442. 333442. You cross a sinestral female of this genotype, homozygous recessive, to a dextral male that's homozygous dominant. What are the expected genotypes and phenotypes of the offspring? All right, so we have a sinestral female, dextral male. What are the expected phenotypes and genotypes of the offspring? Remember, dextral is dominant. This is 333442. All right, we'll give you another 30 seconds.
242.
Yeah, 242D. All right, let's punch in a number here. Okay. Yeah, overwhelming support for A. That is correct. So all sinestral. What? All sinestral because the female's genotype here is homozygous recessive. Right? That's really the only thing you need to look for. The tricky part again, they're all sinestral even though they have that heterozygous dominant allele. Right? So you would normally think in classical Mendelian genetics they'd be all dextral, but because the genotype of the mother was homozygous recessive, the phenotype of the offspring will look as if it were homozygous recessive. Yes?
So the genotypic ratios, like they stay the same, you cross up the same way, but then the phenotype.
Exactly. So the genotypes are going to follow classic Mendelian genetics, but the phenotypes will not. Right? Which is why it's tricky, right? Because you kind of get this dissociation from what you would normally expect. All right, let's go on to the next violation of Mendelian inheritance, and that is epigenetic inheritance. All right, so epigenetics is usually involving chemical modifications to the DNA directly. The example is often methylation. There are other ways that you can epigenetically affect DNA, right? They're wrapped in histones, nucleosomes that can wrap the DNA more tightly or more loosely. Methylation is the one we will focus on for now. Both of these, though, can alter gene expression. Sometimes it appears this can be inherited. And it is reversible. So these are usually transient modifications. You can put a methylation mark on DNA. You can take the methylation mark off DNA. You are at no point changing the DNA sequence itself. Right, you're just putting a little tag on it or removing a little tag on it. It is usually transient. It can sometimes be inherited, although the mechanism by which that is possible is a huge mystery in genetics at the moment. What do we say when we say it can alter patterns of gene expression? Well, let's say you have a gene sequence here. This would be a gene. This will be a promoter. Turns a gene on or off, or this could be an enhancer, etc. These are gene regulatory elements that determine when and where a gene is expressed, which means that it's made, right? It's made into a protein product in this case. Let's say this one is active. Okay. If you methylate parts of it, we'll draw those as little lollipops, especially promoter regions. That can silence a gene. It can interfere with the binding of proteins to the promoter element. The methylation then means that this gene, which is otherwise expressed, is no longer expressed, right? You now functionally have a loss of function protein, but you have no mutations in the DNA itself, right? You just have these transient modifications to the DNA sequence. Now, I should mention methylation isn't always going to be gene silencing, but it does, for now, we'll just think about it as modifying gene activity in the general sense, right? So you have a gene that's now behaving as if it has a mutation, even though there is no change to the DNA sequence. Does that make sense? You'll probably get more into detail in sort of the mechanistic parts of this in the molecular section of the course later in the semester. I will say that this transgenerational epigenetic inheritance is a very controversial frontier in genetics. There's been a lot of really interesting studies where survivors of famine have consistent changes to DNA methylation, and not necessarily the survivors. The grandchildren of survivors of famine sometimes carry somewhat consistent or measurably different ratios of methylation marks in their genome. And in some cases, they are associated with metabolic distress, right? So you can imagine someone going through severe famine, having children. The severe famine, of course, imparts a lot of metabolic distress. The subsequent generations then having separate metabolic distress, in this case associated with abundance of food when they're prepared to have very little food from the famine. Very interesting area. It is circumstantial at best in humans, but it is a thing. There's also, there are situations where in the womb you might be exposed to certain things through your mother. That can lead to methylation marks that can stick with you into adulthood, right? So this idea, again, that exposure to the environment can influence transient modifications to DNA, and those can be lasting, though are also reversible. In the context of epigenetic inheritance, it's really not clear how those methylation marks make it to the next generation. There's some evidence that, as sperm travels through on its way out, it encounters small RNAs that can modify the epigenome. There's also, obviously a lot that can happen in eggs, but I'm mostly pointing out these headlines and articles just to draw your attention to the fact that epigenetic inheritance is important, but also somewhat mysterious, even still in modern biology. There's an area of epigenetics that we are going to talk about a little bit more in depth, and that is going to be dosage compensation. Okay. So the question then is, how is gene expression on sex chromosomes regulated? All right, so in placental mammals, such as ourselves, the males have XY, the females have XX, which means the males are kind of getting gypped. Males just have one X chromosome, females have two, right? So how can we make this more fair and equitable, right? Do the females maybe just turn off one of their X chromosomes, or maybe the male could make twice as many genes on their X chromosome? The important thing to remember, genes on X chromosome, not all specific to sex-related traits. They're just generally important genes, a lot of them. So if males have half as many genes, they have to do something about that, right? They either have to make more of it, or have some ways of sort of compensating for the fact that they don't have those two chromosomes. Well, in placental mammals, it's usually the case that females, in fact, have one X chromosome randomly inactivated. As a result, in any given cell, in someone in the room with two X chromosomes, you're only making genes products from one of those X chromosomes, which means that males can have the same dosage of those genes as females. But that's not how it has to be. In Drosophila, which are fruit flies, they also have an XY system. And in Drosophila, rather than having one of the X chromosomes silenced, the male X chromosome has twofold the amount of expression of those genes as in females, right? So females have two copies of the gene, each making a certain amount of protein. the male is just making twice as much of the protein on the single copy of the gene that they do have, right? So as is common in evolution, there are multiple solutions to this problem of balancing the amount of protein product being made depending on how many copies of a gene that you have. In C. elegans, which is a nematode worm, the males have 2 X chromosomes. The females have an XO system. So the females only have 1 X chromosome. They don't bother with the Y. The males have 2 X. They have a situation where, to compensate, there is a 50% reduction in both X chromosome gene expression. Right? So there's many ways you can go about it. You can turn off an X and then it's equal. You can make the X twice as active and then it's equal. Or you can take the 2 X's and bring each down by 50%. Right? The important thing to remember, though, it is a problem and it is being addressed whenever it arises. Right? So there are many ways to accomplish this task, and I wouldn't remember all of them. But it is being addressed by the cells, right? This gene balance thing is actually quite important. We are going to take a deeper dive into X inactivation, since we are ourselves mammals. Okay, so this will be X inactivation. So here we have a situation where one of 2 X chromosomes is going to get condensed into something called a bar body. Sounds like you're spending too much time at the pub. And that one is silenced. Okay, so the one that gets condensed to the bar body is silenced. This happens randomly, as in terms of which X chromosome, and mid-development. Okay. And because it happens randomly, you have 2 X chromosomes, one of the 2 will randomly get silenced. And because it's happening mid-development, as you already have a good number of cells, it's going to lead to a patchwork phenotype, commonly seen in calico cats, so when you see calico cats or sort of irregular spotting patterns like this, it's often related to random inactivation during mid-development. Let's actually draw that out, which I think will be helpful in illustrating what is happening here. So let's say we have an embryo with 4 cells, and X inactivation actually happens much later than this. Okay, and let's make a black and a, oh dear. Oh no, that's fine. We'll make a black chromosome and a gray chromosome. This is more of a white than anything else, right? So at some point in development, you have 2 X chromosomes, one with a black pigment allele and one with a mutant version that is, let's say, recessive. I feel like I lost my pencil. I don't care for that one. Okay. So let's give this one a dominant B allele and this one a recessive B allele. This one will have a dominant B. This will have a recessive, dominant, recessive, dominant, recessive. Okay. The B allele here will be black pigment. Okay, and the recessive allele will be white or no pigment. Okay. What happens is this embryo has both alleles. Both are being made potentially if it was making pigment, right? You could have the black and the white. Later on, though, don't like. I don't know where that went. All right, sorry about that. Okay, so in this case, we'll have 4 cells again. But let's say X inactivation happens. That will give us a bar body some of the time, and it'll be random which chromosome is being inactivated, right? So we can keep the recessive B active as is happening here or here, or we could turn that one into a bar body and only have the black allele, right? So this cell is going to make no pigment. This cell is going to make black pigment. Okay, these are bar bodies. And what you're seeing is random inactivation of a chromosome. Okay, so we start out with a chromosome that will make black pigment and one that will make no pigment. We've randomly turned off one of those 2 chromosomes, leaving us with either only the black dominant allele or the white recessive allele. This randomness means that the cells that happen to turn off the white chromosome will be black, and the cells that happen to turn off the black chromosome will be white. Yes?
If it's random, does it happen in your equal probability? Like the same?
You would expect if you summed all the cells together in the body that it would be equal. It may be, so this is going to happen at a point in development where the subsequent cells will continue dividing, and so it could be that this cell has more daughter cells than this cell does just because of where it is in the embryo, right? So in that sense, it might not be completely random, but it is otherwise random. I don't know if that answered your question or not. Kind of. But you have a straight 50% chance of either chromosome, and that should be equal. But whether or not it ends up being equal depends on when and where it's happening in the embryo. So what's kind of remarkable about this, in my opinion, is a couple things. One, if you are in the room and have 2 X chromosomes, depending on what part of your body we're talking about, you're either making the one from your mom or the one from your dad, but never both. And so if you have a phenotype from an allele on your father, it may exist in certain parts of your body, and the rest of your cells may not have that phenotype, which I think is kind of interesting. So you're a mosaic, even though it's not as obvious. It's obvious in the calico cat because it's pigment, right? In humans, where it isn't pigment, you just don't see it, but it's happening there anyway. So if you had pigments like calico cats, we would also look like calico cats, which I think is kind of cool. Another kind of cool thing, cells can count their X chromosomes. So there are many disorders where there are extra X chromosomes, but always only one will be active. So in a normal female, 2 X chromosomes, 1 bar body, one of them gets inactivated. Normal male, 1 X chromosome only, right? No bar bodies, right? Because they already have 1 active X chromosome. Turner syndrome, which is phenotypically female, have an XO system, so no Y chromosome. They don't have any bar bodies because they only had the 1 X chromosome. Here's where I think it gets interesting. Triple X, 3 X chromosomes, 2 bar bodies. So even if there's 3 X chromosomes, the cell keeps only one of them active somehow. Right, so the cells are able to count the number of chromosomes. Klinefelter syndrome, phenotypically male, XXY, will have a bar body, right? So males don't usually have bar bodies because they only have 1 X chromosome. So at the end, the cells are somehow only keeping 1 X chromosome active, no matter how many X chromosomes there are, which I think is kind of interesting. Because of this X inactivation, and because calico is basically a heterozygous condition, let's do an example with calico cats and inheritance probabilities for an attendance question. This is session ID 112070, 112070, 112070. So we have a calico cat female. Calico cat is going to be heterozygous for a dominant B allele that makes pigment and a recessive allele that would give orange coloring. They mate with an orange male, has the recessive B allele on the X chromosome. What was the phenotype ratio of their progeny? 112070. This is for attendance, so any answer will do here. Let's do another 20 seconds. All right, please put in an answer if you haven't already. Somehow more of you have put in an answer. Okay. Yes, overwhelming support for A. We'll go ahead and write that out, which is one orange female, one calico female, one black male, one orange male. So how do we get that situation? So we have an orange male, something like that. We have a calico female, which is going to be heterozygous. Remember, this is orange, this is black. You get orange and black spots because one of these two is randomly inactivated. So if we do this Punnett square, we have a calico female here, but we also have an orange female here, right? One of these X's is still getting inactivated, but you have only orange alleles, so it doesn't really matter in terms of fur color. Then we have a black male cat and an orange male cat, right? So one of each, a lot of different possibilities because of this X inactivation. Does this make sense so far? Okay, good. We have one more to do, and that is extranuclear inheritance. So write that out. All right. This is also known as cytoplasmic inheritance. Okay, cytoplasmic. And it's caused by genes being found in mitochondria or chloroplasts, depending on if you are a plant or not. So mitochondria, as you recall, look exactly like that. They have inside of them a circular genome all to themselves. This is ultimately a remnant of when mitochondria were free-living organisms, single-celled organisms. If you recall from biology, there's this something like that. Endosymbiosis theory, right? The big cell eats a smaller cell, right? You've got a little bacteria living inside now. At somehow, some way, that bacteria was the ancestor to all mitochondria. Over time, it lost its ability to live independently. It lost many of the genes it needs to survive. Those have made their way on to the nuclear genome, but it retains its own circular genome. It's about 17,000 base pairs long, and it contains, at this point, 13 genes, not too many, as well as genes for tRNA and R ribosomal RNA. Right, so for whatever reason, not everything made its way to the nuclear genome. Some of those genes still remain in the mitochondria, which means if you have alleles of those genes that have a phenotype, you will inherit them directly from the mitochondria and have without any contribution from the nuclear genome. We also have chloroplasts depicted here pretty well. The chloroplast genome is the same situation as mitochondria, by the way, is about 10 times the size of the mitochondrial genome. For example, tobacco is not how you spell tobacco, is about 156,000 base pairs, right? So much, much bigger than the mitochondrial genome. It contains, depending on the species, about 110 to 120 genes, as well as tRNA and ribosomal RNA. Right, so chloroplasts and plants in general, a lot of variation in chloroplast genome size, a lot of variation in chloroplasts, a lot of variation in general in plants. Mitochondrial genome a little bit less, more or less 13 genes across species. Now, as far as inheritance is concerned, there's a couple things to keep in mind. One is that the egg has around 100,000 times more mitochondria than sperm. Okay, which means that effectively all of your mitochondria come from the mother. Okay, and this is true in at least animals. There are cases where that inverts. So it's not one of the reasons this is confused with maternal inheritance is because this is entirely coming from the mother. The difference with this and maternal inheritance is that when we talk about maternal effect, we are talking about a transient thing happening in the embryos while you're only relying on mom's genes. That then wears off as the zygote starts making its own genes. When we are talking about cytoplasmic inheritance, we are talking about the fact that mom is giving you all of your mitochondria for life, and that will continue affecting you forever, right? So you're not going to get anything from dad here. You're going to be relying solely on what's coming from the cytoplasm in this case. Cells can have more than one organelle variant. And if they do, it is called heteroplasmy. So just as you can have two copies of you have two copies of a gene, you could have dominant recessive, you could have multiple alleles, right? You could have more than one organelle variant in the many thousands of organelles that live within every single cell, right? So rather than just having two chromosomes now, you have a whole soup of mitochondria that are not necessarily clonally identical to one another. They themselves have their own variation. And if you have variation in those organelles, it is heteroplasmy. There's an interesting phenotypic consequence of this, heteroplasmy, which we will get into here. Right, cells, it's redefining it here. can have more than one organelle variant. I don't like this pencil I'm using. And that comes into play in a more conspicuous way, not unlike the calico cat, in variegation of plant leaves. So let's go ahead and draw that out. So you can have a plant leaf here that has pockets of rich green pigment and pockets of white pigment, right? As I'm sure we've all seen when we've looked at variegated plants. This is a variegated leaf. Okay, what's happening is if you look at the white patches, you have cells that mostly have an organelle variant, a chloroplast variant in this case, that is itself relatively pale. And if you look at the green patches that are dark, you have cells that have mostly a darker colored chloroplast. Remember, chloroplasts are giving that green color to the plants. They can be like this, where they have a entirely one type, or it can be sort of an intermediate that contains a mixture of the darker chloroplast and the lighter chloroplast. But that's basically what's happening in a variegated plant, is that certain cells have more or less of the two chloroplast variants, the one that's either really dark green or the one that's light. So how do you end up with a situation where a adult organ like a leaf has a mixture like this? Wouldn't that be equal in every cell? Well, it turns out that is not the case, and that's mostly due to random chance. So if we have a cell here, and there will be two variants, two variant chloroplasts. Okay, we've got a dark one and a lighter colored variant. Okay. That cell is going to randomly divide. And when it does, it's just going to take with it, as it divides in two, whatever part of the cytoplasm happened to be on that side of the cell. Right? So you've got a cell filled with, in this case, light and dark chloroplasts. When it divides, it can be a situation where by random chance, one of the cells ends up with more green in it that's dark and less light chloroplasts. And one daughter cell could end up randomly with more light chloroplasts and less green chloroplasts. Right? You have these chloroplasts that are randomly in the cell divides. Depending on the random positioning of those chloroplasts as the cell is dividing, you can end up with a daughter cell that has more green or a daughter cell that has more white. And this can then happen over and over again, giving you chloroplasts that are mostly green or chloroplasts that are mostly white. Right? And this is happening within an individual, right? This is, again, conspicuous when we're talking about chloroplasts and leaf color, but it's also happening within our own bodies, where certain parts of our body may have more organelle variants of one type than another, just because of the random asymmetric cell divisions, where these organelles, which are just floating in there, are passively passed to different daughter cells. Does that make sense? Okay. Let's go through some inheritance examples with the leaf to see how this could all shake out. Let's say we have, and we're going to pretend that leaves in this case follow inheritance patterns just like we're used to, where the female is providing, in this case, all of the chloroplasts. That isn't universally true in plants, though. All right, so we have a green leaf and a white leaf. The white leaf is female. They come together and they make a new plant. What do you think the color of that leaf is going to be in that generation? Any ideas? White. It'll be all white because in this case, the female is the one providing all of the chloroplasts. So it doesn't matter what the male had. kind of like maternal effect. You are getting only the white leaf, so these are going to be all white. Okay, let's do another example. We'll actually do it down here. Oh my gosh, okay. We're actually going to do 4 total. Let's say we have a green female, and we'll cross that to an all-white male. What do we think that'll be? All green. That is correct, because again, the female is the one providing the chloroplasts here. Right, so this will be female, male. This will be all green. All right, now here's where it gets kind of interesting. Let's say we have a variegated female, right? So it'll be a little bit more patchy. And we have a green male plant. Okay, look at that. All right, so female's variegated. The male is all green. What do we think we could have here? Right, you could have everything. Remember that asymmetric cell division that's happening here can also happen during oogenesis. So you could have eggs that are entirely one organelle or entirely another organelle, and they could be different from one another. Or you could inherit both copies, giving you all green, variegated, and all white. Right, so we have all possibilities here. Right, distinguishing itself again from maternal effect, right? Maternal effect would be just about the genotype of the mother. This is a situation where just random passages of chloroplasts can determine multiple fates. Let's do one more example. Let's say we have a variegated male and a green female. What do we think we have here? All green, that's right. That is all green. Because again, it only matters what the female, the one providing the chloroplasts, is doing. Does this make sense? Okay, good. I think it'll become more interesting to many of you if we then close by talking about human mitochondrial disease. Okay. So there are more than 200 known diseases that in some way affect mitochondria. Kind of makes sense, right? If mitochondria is making all the ATP, it's therefore super important in producing energy in a lot of fundamental cellular processes. It would stand to reason that anything that messes with mitochondrial function could cause some amount of disease. You will often find that it involves ATP-rich tissues, at least the phenotypes, right? Things like neurons or muscle. These are going to be more affected by a mitochondrial mutation than tissues that don't need a lot of energy, right? You can maybe limp by without a lot of mitochondria, but if you're a neuron that's constantly firing, constantly needing lots of energy, then you're more likely to see a phenotype that's sort of neuromuscular than you are something in like the skin or something perhaps a little less active. You will often see then, if it is on a mitochondrial genome, a maternal inheritance pattern. If you're looking at like a pedigree, for example. Because again, mom has all the mitochondria, right? So if you have a mitochondrial disorder, you probably got it from your mom. And not from your dad, who's contributing very little, as always. heteroplasmy that we see is a situation that leads itself, as you imagine, with sort of the variegated plant leaf, right? If you are a variegated plant leaf, but you swap variegation with a mitochondrial disease, you have patches of healthy tissue and passes of mitochondrial, regions of mitochondrial disease, right? So you get a wide phenotype spectrum, even within families. So you could have a situation where, a mother and a daughter have the same mitochondrial variant, have totally different phenotype spectrums, because in the mom, random cell divisions are asymmetrically mixing and matching mitochondria around, right? Mom might have more diseased mitochondria in neurons, and the daughter might have more mitochondrial mutations in muscle, right? So maybe one has neurological symptoms, the other has weakness, right? But it's going to be variable because it's just that randomness of those cell divisions, right? Whatever happened, however they happen to sort of segregate out during cell divisions, can give you really wide phenotypic variants in general. Here are some examples for those that are interested. Many of them, as you'll see, again, neurogenic muscle weakness, neuropathy, myopathy, cardiomyopathy, right? A lot of these are going to be involving muscles and neurons, which again use a lot of ATP. And again, you may have avoided any diseased mitochondria in your heart just by random chance, right? And so an individual with the same disorder, the same mutation, might have a severe cardiomyopathy. You might have none, but you both have the diseased mitochondria. And that's just this idea that as cells divide in the early embryo, just like with X activation, there's a randomness that ensues that may or may not lead to various disease phenotypes. Otherwise, with that is all I have. There's a summary at the end. There's always a summary at the end. We just never get to it. If you have any questions, let me know. We'll post the exams hopefully sometime today.